When I look at this, and these are 3 billion chemical letters, instructions for a human being, my eyes glaze over. But when scientist Eric Lander looks at this, he sees stories.
The genome is a storybook that's been edited for a couple billion years. And you couldn't take it to bed like 1001 Arabian Nights and read a different story in the genome every night.
This is the story of one of the greatest scientific adventures ever. And at the heart of it is a small, very powerful molecule, DNA. For the past 10 years, scientists all over the world have been painstakingly trying to read the tiny instructions buried inside our DNA. And now, finally, the human genome has been decoded.
We're at the moment the scientists wait for. This is what we wanted to do. We're now examining and interpreting the genetic code.
This is the ultimate imaginable thing that one could do scientifically, is to go and look at our own instruction book and then try to figure out what it's telling us.
And what it's telling us is so surprising and so strange and so unexpected. 50% of the genes in a banana are in us?
How different are you from a banana?
I feel, and I feel I can say this with some authority, very different from a banana.
You may feel different.
I eat a banana.
All the machinery for replicating your DNA, all the machinery for controlling the cell cycle, the cell surface, for making nutrients, all that's the same.
So what does any of this information have to do with you or me? Perhaps more than we could possibly imagine. Which one of us will get cancer, or arthritis, or Alzheimer's? Will there be cures? Will parents in the future be able to determine their children's genetic destinies?
We've opened a box here that has got a huge amount of valuable information. It is the key to understanding disease and, in the long run, to curing disease. But having opened it, we're also going to be very uncomfortable with that information for some time to come.
To begin, let's go back 4 and some billion years ago to wherever it was that the first speck of life appeared on Earth, maybe on the warm surface of a bubble. That speck did something that has gone on uninterrupted ever since. It wrote a message. It was a chemical message that it passed to its children, which then passed it on to its children and to its children and so on. The message has passed from the very first organism all the way down through time to you and me. Like a continuous thread through all living things. It's more elaborate now, of course, but that message, very simply, is the secret of life. And here is that message contained in this stunning little constellation of chemicals we call DNA. You've seen it in this form, the classic double helix, but since we're going to be spending a lot of time talking about DNA, I wondered, what does it look like when it's raw, in real life? So I asked an expert.
I mean, DNA has a reputation for being such a mystical, highfalutin sort of molecule, all this information, your future, your heredity. It's actually goop. See, this here is DNA.
Professor Eric Lander is a geneticist at MIT's Whitehead Institute.
It's very, very long strands of molecules, these double helices of DNA, which when you get them all together, just look like little threads of cotton.
And these strands were literally pulled from cells, blood cells, or maybe skin cells of a human being.
Whoever contributed this DNA, you can tell from this whether or not they might be at early risk for Alzheimer's disease. You can tell whether or not they might be at early risk for breast cancer. And there's probably about 2,000 other things you can tell that we don't know how to tell yet, but we'll be able to tell. And it's really incredibly unlikely that you could tell all that from this. But that's DNA for you. That apparently is the secret of life, just hanging off there on the tube.
And already DNA has told us things that no one had expected. It turns out that human beings have only twice as many genes as a fruit fly. Now, how can that be? We are such complex and magnificent creatures, and fruit flies, well, they're fruit flies. DNA also tells us that we are more closely related to worms and to yeast than most of us would ever have imagined. But how do you read what's inside a molecule? Well, if it's DNA, if you turn it so you can look at it from just the right angle, you will see in the middle what look like steps in a ladder. Each step is made up of two chemicals, cytosine and guanine, or thymine and adenine. They come always in pairs called base pairs, either C and G or T and A for short. This is, step by step, a code, 3 billion steps long, the formula for a human being.
We're all familiar with this thing. This shape is very familiar.
Double helix, yeah.
Double helix. First of all, I'm wondering, this is my version of a DNA molecule. Is this, by the way, what it looks like?
Well, give or take. I mean, cartoon version of it.
Cartoon
Yeah.
version.
You know, a little like that or so, yeah.
So there are, in almost every cell in your body, if you look deep enough, you will find this chain here.
Oh, yes, stuck in the nucleus of your cell.
Now, how small is this? If in a real DNA molecule, the distance between the two walls is how wide?
Oh, golly.
Look at this, he's asking for help.
This distance is about, this distance about 10 angstroms, which is...
That's 1/1000th of a meter when it's clumped up in a very particular way.
Well, no, it's curled up some like that, but you see, it's more than that. You can't curl it up too much because these little negatively charged things will repel each other. So you fold it on its... I'm going to break your model.
don't break my model.
Yeah, I don't want to break your
Very
model. But,
valuable.
you got this, and then it's folded up like this, and then those are folded up on top of each other. And so, in fact, if you were to stretch out all of the DNA, it would run, oh, I don't know, thousands and thousands of feet.
Okay. But the main thing about this is the ladder, is the steps of this ladder. If I knew it was A and T and C and C and G and G and A...
No, it's not G and G,
I'm
it's
sorry,
G and
whatever
C.
the rules
You got
are
the
of the
base
grammar.
pairs, yeah, right.
If I could read each of the individual ladders, I might find the picture of what?
Of your children. This
Of
is
my
what you
children.
pass to your children. Oh. People have known for 2,000 years that your kids look a lot like you. Yeah. Well, it's because you must pass them something, some instructions that give them the eyes they have and the hair color they have and the nose shape they do. The only way you pass it to them is in these sentences. That's it.
And to show you the true power of this molecule, we're going to start with one atom deep inside. We pull back, and you see it form its A's and T's and C's and G's and the classic double spiral. And then it starts the mysterious process that creates a healthy new baby. And the interesting thing is that every human baby, every baby born, is 99.9% identical in its genetic code to every other baby. So the tiniest differences in our genes can be hugely important, can contribute to differences in height, physique, maybe even talents, aptitudes. and can also explain what can break, what can make us sick. Cracking the code of those minuscule differences in DNA that influence health and illness is what the Human Genome Project is all about. Because sometimes, out of 3 billion base pairs in our DNA, just one single letter can make a difference. Allison and Tim Lord are parents of 2-year-old Hayden.
Hi, Pumpkin.
The two things that I think of the most about Hayden, which a lot of people got from him right from the beginning, is that he was always, I thought, very funny.
Make your very, 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 very serious face.
I mean, he loved to smile and laugh, and he just used to guffaw. I mean, this was later when he was about 1 year old. He just found the funniest things hilarious, and so he and I would just crack each other up.
Peekaboo!
Hayden seemed to be developing normally for the first few months, but Allison began to notice that some things were not quite right.
I was very anxious all the time with Hayden. I sensed that something was not the same. I would see my friends changing the diaper of their child who was around the same age, their newborn, and see the physical movement and the legs moving and things like that, and Hayden didn't do that.
Happy birthday to you.
Doctors told them that Hayden was just developing a bit slowly, but by the time he turned 1 year old, it was clear something serious was wrong. He never crawled, he never talked, he never ate with his fingers, and he seemed to be going backwards, not progressing.
I remember the last time he laughed.
Yeah.
And I took a trip with him out to pick up a suit, because we were going to a wedding that night, and we came back, and it was really windy, and he just loves to feel the wind, and so we had a great time. I came back, and I propped him up right here on the couch, and I was sitting next to him, and he just kind of threw his head back and laughed, like, what a fun trip, And that's the last time he was able to laugh. It's really hard.
It turned out that Hayden had Tay-Sachs disease, a genetic condition that slowly destroys a baby's brain.
What happens is the child appears normal at birth, and over the course of the first year begins to miss developmental milestones. So at 6 months, a child should be turning over. A child is unable to turn over, to sit up, to stand, to walk, to talk.
Tay-Sachs begins at one infinitesimal spot on the DNA ladder when just one letter goes wrong. Say this cluster of atoms is a picture of that letter. A mistake here can come down to just 4 atoms, that's it. But since genes create proteins, that error creates a problem in this protein, which is supposed to dissolve fat in the brain. So now the protein doesn't work, so fat builds up, swells the brain, and eventually strangles and crushes critical brain cells. And all of this is the result of one bad letter in that baby's DNA.
In most cases, it's a single base change, as we say, a letter difference.
One defective letter out of 3 billion, and no way to fix it.
That's my boy.
Tay-Sachs is a relentlessly progressive disease. In the years since his diagnosis, Hayden has gone blind. He can't eat solid food. It's harder and harder for him to swallow. He can't move on his own at all. And he has seizures as often as 10 times a day.
For children with classical Tay-Sachs disease, there's only one outcome, and children die by age 5 to 7, sometimes even before age 5.
As it happens, Tim Lord has an identical twin brother. When Hayden was diagnosed, that brother Charlie went to New York to be with Tim. And of course, Charlie called his wife, Blythe, to tell her the news. Blythe had been Allison's roommate in college and her best friend.
What are you going to say about that?
Charlie told me that Hayden had Tay-Sachs. He called me on the phone and he told me immediately what it was. I went up onto the computer and looked it up and then just couldn't believe what I read.
So you finish your cookie?
Blythe and Charlie had a 3-year-old daughter, Taylor, and a baby girl named Cameron. Cameron was healthy and happy except for one small thing.
On the NTSAD website, it talks about typically between 6 and 8 months is when the signs start coming, but one of the early signs is that they startle easily. And Hayden had always had a really heavy startle response. Well, we'd noticed that Cameron had a comparable startle response, not quite as severe, but absolutely not like Taylor had had.
As soon as she saw that early warning sign on the Tay-Sachs website, Blythe went to get herself and Cameron tested.
It was another week. It was exactly a week until we got the final results on Cameron's blood work. And then the Tuesday before Thanksgiving, we went into our pediatrician's office and he had the results, and we found out that night that Blythe was a carrier and that
Cameron had
Tay-Sachs.
He said, all I said was, I'm sorry.
Tay-Sachs is a very rare condition. It usually occurs in specific groups, like Ashkenazi Jews, and even then, the baby must inherit the bad gene from both parents. So even though there is a Tay-Sachs test, the Lords had no reason to think they would be at risk. And yet, incredibly, all 4 of them, Tim and Charlie and both their wives, all 4, were carriers. That was an unbelievably bad roll of the genetic dice.
Hey, little nugget.
Charlie and I are incredibly close and have been all our lives.
Yeah.
And when I think about him and Blythe having to go through this, it just seems really cruel. It just seems too much.
I had already geared myself up for being my brother's rock, and I couldn't imagine having to help him and go through it myself.
Here's Blythe.
For families like the Lords, and for everybody, the Human Genome Project offers the chance to find out early if we're at risk for all kinds of diseases.
I love Taylor.
I would like to see a really aggressive push to develop a test for hundreds of genetic diseases so that parents could be informed before they started to have children as to the dangers that face them. And I think it's within our grasp. Now that they've mapped the human gene, I mean, the information is there for people to begin to sort through. They're horrible, horrible, horrible diseases. And if there's any way that you can be tested for a whole host of them and not have them affect a child, I think it's something that we have to focus on.
Hayden Lord died a few months before his 3rd birthday.
Curing people is the whole point, right? But if there is one thing that the Human Genome Project has taught us, it's that finding cures is a whole lot harder than simply reading letters of DNA. Take, for example, the case of little Riley Dimanche.
At 2 months, Riley appears to be a perfectly healthy baby boy, but he's not. When Riley was just 13 days old, Kathy Dimanche got the call that every parent dreads.
My pediatrician called on a Thursday evening, and he said, I need to talk to you about the baby. He said, are you sitting down? And I'm like, yeah, and that really surprised me. And he said, are you holding the baby? Because he didn't want me to drop the baby, obviously. And he said that tests came through and Riley tested positive to cystic fibrosis. And I was in shock.
As Kathy and her husband would soon learn, cystic fibrosis, CF for short, attacks several organs of the body, but especially the lungs. Its victims suffer from chronic respiratory infections, and half of all CF patients die before the age of 30.
Sounds good.
Good.
Sounds great. I think we can still be hopeful that their child will grow up to have a normal, healthy, happy, and long life. But at the present time, I don't have any guarantees about that.
Someone had asked me, are you prepared to bury your son at such a young age, whether it's 4 or 40? And he was 17 days old when that happened. And I said, I've had him for 17 days. I wouldn't trade those 17 days.
Finding the genetic defect that causes CF was big news back in 1989.
Medical researchers say they have discovered the gene which is responsible for cystic fibrosis, the most common inherited fatal disease in this country.
We're going to cure this disease.
A lot of people expected the cure to arrive any day. It didn't.
Cystic fibrosis.
Francis Collins, now head of the government's Genome Project, led one of the teams that discovered the CF gene.
We still have not seen this disease cured or even particularly benefited by all of this wonderful molecular biology. CF is still treated pretty much the way it was 10 years ago, but that is going to change.
The original hope was that babies like Riley could be cured by gene therapy, medicine that would provide a good working copy of a broken gene. But attempts at gene therapy have hardly ever worked. They remain highly controversial, so if there's going to be an effective treatment for Riley, instead of fixing his genes, we're going to take a look, and this is new territory, at his proteins.
What do proteins do? When you look at yourself in the mirror, you don't see DNA, you don't see RNA. You see proteins and the result of protein action. That's what we are physically composed of.
So it's not a Rodgers and Hammerstein thing where one guy does the tune and the other guy does the lyrics. This is a case where the genes create the proteins and the proteins create us.
That's right. We are the accumulation of our proteins and our protein activities.
A protein starts out as a long chain of different chemicals, amino acids, but unlike genes, proteins won't work in a straight line.
Genes are effectively one-dimensional. If you write down the sequence of A, C, G, and T, that's kind of what you need to know about that gene. But proteins are three-dimensional. They have to be, because we're three-dimensional and we're made of those proteins. Otherwise, we'd all sort of be linear, unimaginably weird creatures.
Here's part of a protein. Think of them as tangles of ribbon. They come in any number of different shapes. They can look like this, or like this, or this. The varieties are endless. But when it's created, every protein is told, here is your shape. And that shape defines what it does, tells all the other proteins what it does, and that's how they recognize each other when they hook up and do business. In the protein world, your shape is your destiny.
They have needs and reasons to want to be snuggled up against each other in a particular way. And actually, a particular amino acid sequence will almost always fold in a precise way.
Should I think origami-like? I mean, should I think folding and then...
It's very elegant, very complicated, and we still do not have the ability to precisely predict how that's going to work. But obviously it does work.
Except, of course, if something does go wrong. And that's what happened to baby Riley. Riley has a tiny error in his DNA. Just 3 letters out of 3 billion are missing. But because of that error, he has a faulty gene, And that faulty gene creates a faulty or misshapen protein. And just the slightest little changes in shape and boom, the consequences are huge. Because it is now misshapen, and a key protein that's found in lung cells, in fact in many cells, can't do its job. So let's take a look at some real lung cells we'll travel in. This is the lining or the membrane of a lung cell, and here's how the protein is supposed to work. The top of your screen is the outside of a cell, the bottom of the screen the inside of the cell, of course, and our healthy protein is providing a kind of chute so that salt can enter and leave the cell. Those little green bubbles, that's salt. And as you see here, the salt is getting through. But if the protein is not the right shape, then it's not allowed into the membrane. It can't do its job. And without that protein, as you see here, salt gets trapped inside the cell, and that triggers a whole chain of reactions that makes the cell surface sticky and covered with thick mucus.
The first two positions that are done sitting up are probably a little more difficult to do.
That mucus has to be dislodged physically.
Just kind of like that.
Riley's family is learning to loosen the mucus that may develop in his lungs and fight infections with antibiotics.
You want to sort of do it with a cupped hand.
Trying to get at the top of the lungs.
Yeah, you want to be like right.
But what the doctors and the scientists would love to do is if they can't fix baby Riley's genes, then maybe there's some way to treat Riley's misshapen protein and restore the original shape. Because if you could just get them shaped right, the protein should become instantly recognizable to other proteins and get back to business. But look at these things. How would we ever learn to properly fold wildly multidimensional proteins? It may be doable, but it won't be easy.
The Genome Project was a piece of cake compared to most other things because genetic information is linear. It goes in a simple line up and down the chromosome. Once you start talking about the three-dimensional shapes into which protein change can fold and how they can stick to each other in many different ways to do things, or the ways in which cells can interact, like wiring up in your brain, you're not in a one-dimensional problem anymore. You're not in Kansas anymore.
Okay, so it's not just the number of genes, it's all the different proteins they can make and then the way those proteins interact. And to figure out all those interactions and how they affect health and disease, that's going to keep scientists very busy for the next few decades.